The most important things that's happen in the third week of development is formation of the trilaminar gem discs. This process is happen by one important mechanism that name is a gastrulation. It means that the gastrulation is a process by which that inner cell mass is converted into a trilaminar embryonic discs. The most characteristics for the gastrulation is formation of the one streaks on the surface of the epiblast layer. You can see here the primitive streak. In the cephalic end of the primitive streak, you can see that the one depression, that name is a primitive pit. This depression is surrounded by small elevated epiblast cells that name is a primitive nodes. Here you can see again the primitive streak, primitive pit, and primitive node. When the streak is formed, the epiblast cells around the streak started to movement toward the streak, and they wanted to go down through this streak. This mechanism and going down the cells to underlying surface is name is invagination. For the invagination of the epiblast cells, we have to doing some changes in the epiblast layer. The most important things is downregulation of the E-cadherins protein between the epiblast cells. When the E-cadherin is downregulated, the attachment of the epiblast cells is loses and they attach from the basic membrane, and this epiblast cells be a flask shape and going down and invaginated through the streak. The first of all, that cells migrated through the primitive streaks, they displace the hypoblast and creating the embryonic endoderms here. The number 4, the definitive endoderms. The second wave of the epiblast cells that migrated through the streaks, making a new layer between the new endoderms and epiblast cells. Here, these cells, his name is a intraembryonic mesoderms. When the intraembryonic mesoderm is formed, the primitive streaks gradually disappear and is closed in the end of the third weeks. The remaining epiblast cells change and making a ectodermal layers. In conclusions, all three germs layer, it means that endoderms, intraembryonic mesoderms, and ectoderms are formed by the epiblast cells. The cell migration and specification through the primitive streaks are controlled by one growth factor, that name is fibroblast growth factor number 8, which is synthesized by streak cells themselves. This growth factor controls movement by downregulation E-cadherin, a protein that normally binds epiblast cells together. Fibroblast growth factor number 8 then controls cell specification into the mesoderm by regulation of the one protein that name is a brachyury. Another thing that's formed during the gastrulation is name is a form notochords. Notochord is the one process that's important for the formation of the neural tubes and the spinal columns. Here again in this picture you can see the pathway of the migration of the primitive streaks, and you can see that some cells migrated through the primitive pits, and I will talk about these cells that migrated directly from the primitive pits. Here I want to tell you about the one points that the loose network of tissue called mesenchyme or embryonic connective tissue that appear after the following of the formation of the primitive streak. And usually in embryology, when we talk about the mesenchyme, it means that embryonic connective tissue. This is one term. Another things that you can see in here is a real pictures that you can see that the primitive streaks here, the primitive groove and streaks, and this is a primitive pit. You can see clear in the real picture. Again, that this picture show that the formation of the notochord. As I told you, the one mechanism for the formation of notochord is happen when that epiblast cells migrated through directly from the floor of the primitive pits. That cell then migrated from the primitive pits. The first of all, the first wave of the cells making a prochordal plates, and the second wave that cells migrated through the pits making the notochords. Here the pictures show that the formation of the precaudal plate and the notochord. As I told you, this is a primitive pits that cells migrated. In the first wave is coming in front of the notochord, and the second wave of the cells that migrated through the pits making the notochords. These structures are important for the formation, the brain, neural tubes, and vertebral columns. The more development of notochord here is shown by this simple picture. Again, the precaudal plates. Another point in this picture, you can see that through depression in the cephalic and the caudal end of the embryo here, this cephalic depression is named is the oropharyngeal membranes. Here, the ectodermal layer directly attached to the endodermal layer. It means that here we don't have the mesoderms. Again, in the caudal end of the embryo, we have a one depression like a oropharyngeal membrane. Here, again, the endodermal layer directly attached to the ectoderm, and we don't have a mesodermal layer because the red color is a mesodermal layer. The name of this depression in the caudal region is name is cloac membrane. By the middle of the third week, intraembryonic mesoderm separates the ectoderm and endoderm except in the cloac membrane and oropharyngeal membrane, as I told you and you saw in the last pictures. Here this electron scanning microscope that show that ectodermal layer, mesodermal layer, and endodermal layer in the trilaminar embryo germ discs. Sometimes the primitive streak function slowed down at the end of the fourth week normally, but sometimes the primitive streak is remain. As a result, we have excess migration through the streak. As a result, the remaining of the primitive streaks after the normal times and excess migration, we have some abnormalities, especially in the sacrococcygeal area. That name is teratoma. And when this form in here, we say that sacrococcygeal teratoma. And when you dissected this teratoma, you can find the trigerm layers, and maybe you find the bone, nerves, and some digestive tissue in here. And as I told you, the notochord is a cellular rod that developed from the notochordal process, but the formation of the notochords is a complex process that I want to tell you in the real pictures. Here, in the mid-sagittal section, the transverse section, again mid-sagittal section, you can see that the notochordal formation, that the real pictures, as I told you, that cells migrated through the primitive pit, go straight toward the oropharyngeal membrane, these cells making the notochord. But during the formation of the notochords, you can see that the first of all, the notochord is it has a one narrow structure, and this narrow structures, the first of all, attached to the endoderm. The yellow color here is the endoderms, and after the short time, the canal it's be opened and making one notochordal plates, and in the next steps, these plates again they attach from the endoderms and finally be a solid tissue. That name is a definitive notochords. It means that the first of all we have a notochordal process, but during the more development be a definitive notochord. This transformation of notochordal process to the definitive notochord take along around the 16 to 22 days of gestation. The notochord is important for the induction of the ectoderm, for the formation of the neural tubes, and induction of the paraxial mesoderm for the formation of the vertebral columns. When these structures are formed, the notochord regress, and finally it persists in adult as a nucleus pulposus in each vertebral discs. During the formation of the notochords, a temporary canal and small canal, that name is a neuroenteric canals, here shown by arrow, connected the amniotic cavity and yolk sac. Following the cessation of the notochord development, neuroenteric canal obliterated. If you want to learn more about the notochord formation, you can see that these pictures by detail and you study by yourselves. Finally, in the third week, you can see that the embryo has three germ layers. The blue color is ectoderms, the yellow color is definitive endoderm, and here the red color is an intraembryonic mesoderm, and here is a notochords. The intraembryonic mesoderms divided in three parts. That part near to notochord and near to midline is name is a paraxial mesoderm. Here this cells in the middle area is name is intermediate mesoderms, and the lateral side of the mesodermal cell is name is a lateral plate mesoderm, and in future developments the lateral plate mesoderm divided to somatic layers and splanchnic layer, and one cavities appear between the somatic and splanchnic cavity, that name is intraembryonic coeloms. You can see here the paraxial mesoderms, intermediate mesoderm, and lateral plate mesoderms in a real embryo. Again, here you can see that the three germ layers, the notochord in the midlines, the formation of the paraxial, intermediate, and lateral plate mesoderm is a mid-sagittal section. You can see that the notochord in here. And this picture shows that the neurulations, and I want to talk about the neurulation after the short break.